At the heart of every brain-computer interface lies a fundamental challenge: how to capture and interpret the electrical signals that represent our thoughts, intentions, and sensory experiences. Now, while all BCIs share this goal, the method that they use in order to access the brain's activity and the neural signals essentially just differ significantly in terms of their precision, safety, and practicality. Now, in our previous video, we looked at this concept of single unit activity, which essentially refers to the neural activity of individual neurons. And in this video, we'll take a closer look at the other three key neural recording modalities, which include local field potentials, also known as LFPs, electrocorticography, and also EEGs, which all essentially just offer a unique window into the brain function and directly support the different classes of BCI applications. And we're first going to start with local field potentials. Now, local field potentials, or LFPs, are essentially a type of extracellular electrophysiological signal that's generated by the synchronized electrical activity of a local population of neurons. And this is not individual neurons, we're talking about a group of neurons here. And rather than capturing the sharp millisecond scale spikes of individual neurons, LFPs essentially just reflect the slower integrated input and local processing within a neural circuit. And we can see how that is by examining this graph right here. Here, the raw signal. This is the central black trace that is a full bandwidth extracellular recording, and it also includes low frequency local field potentials and high frequency action potentials, also represented here as spikes. And this is processed through a filter. Now, when you apply a low pass filter, typically under around 300 hertz, the low fluctuations only remain, and these low waveforms essentially reflect the summed synaptic potentials from the nearby neural populations, and this essentially is what makes up the local field potentials, or the LFPs. On the other hand, when we're trying to apply high-pass filters, filters that are generally above 300 hertz, this isolates fast, sharp spikes, these millisecond scale action potentials of individual neurons, and that's represented right here. So we can see from this sort of diagram that LFP signals essentially just reflect the overall trend of the summed synaptic potentials, as opposed to examining the individual neural activity through spiking activity. Now, something that we really need to know about the working mechanism of producing and capturing LFPs is that, first of all, LFPs just typically arise because of synaptic potentials, not action potentials. And now these synaptic potentials are the result of ion flowing through during the synaptic transmission, primarily within the dendrites of pyramidal neurons. And these potentials are captured by intracortical microelectrodes, which are implanted directly into the brain parenchyma. And these LFPs are essentially just rich informations within frequencies ranging from 1 hertz to 300 hertz, and they're subdivided into delta, theta, alpha, beta, and gamma bands. And the signal integrates the activities from neurons within a radius of a few hundred micrometers to just a few millimeters, essentially. Now, in terms of their applications, LFPs have proven extremely valuable in motor recording, specifically in motor cortex-based prosthetic systems. Now, these systems can enable users with paralysis to control robotic arms or cursors using intended movement signals that are extracted directly from the LFP patterns. And they've also proven quite valuable to deep brain stimulation systems for neurological disorders like Parkinson's disease, where closed-loop stimulation is guided by pathological LFP oscillations. And also in research, LFPs generally provide insight into population-level dynamics, such as phase coupling, oscillatory asynchrony, and also cortical communication. Some of the general advantages of LFP application is that it offers excellent temporal resolution, in that it is able to just extract information and record neural signals in a millisecond scale, and it just generally offers moderate spatial resolution. And these LFPs also tend to be more stable over time compared to just single unit recordings, which make them much more suitable for long-term implants. Some of the general challenges that are encountered by LFP application is that it is highly invasive. We again saw this with having to record single unit activity earlier on for BCIs, in the sense that it requires precise placement of electrodes in target brain regions, and also long-term usage of LFPs can essentially just trigger immune response. And also interpretation can, at some instances, be very complex due to the integrated nature of the signals, and disentangling contributions or information from different neural subtypes and layers may be very difficult.